But before that, we have a few words from Diana Azam, who is a board member and also in charge of our membership working group, who has a message for all members that I hope you will pay attention to.
All right. Good morning, everyone. Thank you, Tony. So I wanted to take a few moments to remind everyone that your membership, to actually pay your membership dues. I really think that I can't stress enough how important it is for each and every one of you to keep your membership active. There are many benefits from being a member. One important one coming up is we're gonna have reduced registration rates for active SFPM members to attend next year's precision medicine meeting in Europe, in Copenhagen, Denmark. So that's one of the benefits you're going to have. And also, as you know, exclusive access to recorded seminars, our directory list. And also, I wanted to mention that, if any of you would like to, contribute to a blog, writing a summary, anything related to FPM, please feel free to email me, email Aaron, any of the board members. We'd be happy to have that published on the website. Even if you have any kind of idea that you would like to discuss with us, we welcome contributions from you as a member, and we would love to have you, we would love to publish that on our website. I also want to say that you have any publications from your lab, feel free to send it to us so we can also post it on the website. I really believe that it's important for us to maintain our membership and also invite our colleagues and friends and really enhance our membership. It's very important for us to maintain that. So I thank you for your commitment and support as an SFPM member.
Great. Thank you very much, Diana. Just remind everyone, the SFPM is you guys. There is not some sort of colossal administrative apparatus in a high rise here in Boston. The SFPM is its membership. And if you have ideas and you want to get things done, it's going to be up to you to do them. But it also means you really do have the ability to influence how we operate going forward. With that, I will turn it over to Juan Montero, one of our other board members, who will introduce today's speaker.
Hey everyone, it's my pleasure to introduce Patricia Pérez-Galán. She graduated at the University of Zaragoza, where she obtained her PhD in 2003. Then she moved to Hospital Clínic here in Barcelona for a first postdoc, followed by a second postdoc at NIH. In 2010, she obtained a tenure track at IDIBAPS, also inside the Hospital Clínic, and since 2019, she leads the group of microenvironment in lymphoma pathogenesis and therapy. The focus of her research is the characterization of the crosstalk between lymphoma cells and its immune microenvironment, with the aim of manipulating these interactions to design novel targeted and personalized therapies using patient-derived systems. So I'm pretty sure that we're going to hear great things and interesting for this audience. I would like to remind you all that after her talk, you can directly ask Patricia the questions. Just unmute yourself. Or otherwise, you can write the questions in the chat and I'll read them out loud. Please, all the members, please mute yourself while Patricia is talking. And yeah, Patricia, the floor is yours. Thank you for accepting the invitation.
Okay. Thank you, Joan, for the invitation, Tony, and thank you for this kind introduction. So today, as John introduced, I'm going to try to share with you the models we have developed in the context of non-Hodgkin lymphoma, the disease we work with. So my idea is to show what we have in terms of description of the model and the utility in the context of drug screening and precision medicine. So non-Hodgkin lymphoma is a heterogeneous disease. Basically, there are composed of tumor B cells that originate the lymphatic system, but then they can extravasate to peripheral blood and even other non-lymphatic systems. These non-Hodgkin lymphoma, they derive from B cells in different stages of differentiation. We look into the lymphoid follicles. So each entity derives from a precise moment of differentiation. And in fact, this is used to classify the entities. Hodgkin lymphoma is among the top 10 more frequent cancers. Inside this, the more common entities, we find follicular lymphoma is one of the models we work with. It's the most common indolent lymphoma. And this is characterized by these follicles. That's why this is termed follicular lymphoma, that are positive for the B cell marker CD20 and also for the BCL2. BCL2 is overexpressed as a result of a hallmark translocation between the BCL2 genes and the immunoglobulins. Besides this translocation, which is not enough to really engage lymphomagenesis, other somatic mutations occur, including epigenetic modifiers, immune receptors, and even the acquisition of N-glycan motifs in the B cell receptor. The immunophenotype is the typical of B cells, together with the expression of germinal center markers such as CD10. Another model we work with, but I'm not going to explain today, is diffuse large B-cell lymphoma. It's the most common aggressive, the most common Hodgkin lymphoma, and it's also aggressive. It's basically the counterpart of follicular, the bad guy, the bad brother of follicular lymphoma. And there is a portion of follicular lymphoma patients that transform to diffuse for the diffuse large B-cell lymphoma. The model we are going to also talk today is mantle cell lymphoma. Mantle cell lymphoma is a tradition, basically, where in the work where I work and in the lymphoid neoplasms program in hospital clinic. It's a rare disease, but it's aggressive in most of the cases. And in this case, the translocation, the hallmark translocation is cyclin D1, leads to cyclin D overexpression based on the fusion with the immunoglobulin genes, and collaborates with other somatic mutations that implies epigenetic modifiers, DNA damaging genes such as ATM or p53, and also the Notch pathway. In terms of immunophenotype, the peculiarity is basically the co-expression of a T cell marker, CD5, together with typical B cell markers, CD10 or CD20 or CD19. In the pathogenesis of non-Hodgkin lymphoma is not only fundamental these somatic alterations and genetic alterations, but also the collaboration of microenvironment that is you all know for other cancers. And in fact, follicular lymphoma is basically probably the best example of dependence of the microenvironment. And it was already in 2004 when all the microarray started, when the group of Lou Stout and NIH, in the context of the lymphoma leukemia profiling project, they discovered that the outcome of these patients were related not with the tumor cell itself, but it depends on the composition of the microenvironment. And in fact, they defined specific signatures that were related with poor prognosis or good prognosis. And in the case of poor prognosis, they were enriched in myeloid cells. In fact, the microenvironment of follicular lymphoma is quite rich, and it also complexifies the recapitulation in vitro. It's integrated by different populations of T cells, both CD4 and CD8, different types of macrophages and stromal cells, and among them, the dendritic cells, follicular dendritic cells are one of the most important, as we will see. In the case of mantle cell lymphoma, the situation is a little bit similar, with high infiltration of B and T cells in the lymph nodes. The stromal cells in this case are mesenchymal stromal cells, and also different populations of tumor-associated macrophages. I mean, in conclusion, all these complex network of cytokines, chemokines, and adhesion molecules, together with these somatic recurrent mutations, so leads to this the origin of these diseases. And this complexity is also is challenging when we want to reproduce in our preclinical models. And this is what we are trying in the last years in our lab. Just to give you an idea of how these diseases are treated, so they are basically treated with chemoimmunotherapy using rituximab, anti-rituximab, or other anti-CD20 as a backbone. Sometimes the chemotherapy is different from the two diseases. And in fact, the introduction of immunotherapy was really a breakthrough in the treatment of these patients. And as you can see here, starting in 2000 is when really the overall survival really improved comparing with the previous years with just the application of chemotherapy. These two diseases are the ones we are going to talk about, are different because, as I said, follicular lymphoma is indolent, and in fact, the overall survival is pretty good. But it's characterized by frequent relapses, and a portion of them, as I said before, transformed to DLBCL, diffuse large B-cell lymphoma, meaning also a very dismal prognosis. In the case of mantle cell, which is by definition aggressive, even though there is a portion of them, about 50%, which have an indolent behavior. So in this case, the patients normally respond very well at the beginning, but they relapse early and very badly. And the problem is to rescue them from this relapse. So in the last years, the introduction of another type of agent, the BTK inhibitors, really have also give light to this disease. But still, those patients relapsing to BTK also have a very dismal prognosis. And it's evident that we need to continue investigating and designing novel therapies. And to do that, we need to have good models. And the novel therapies that we want to assess are all basically in the context of immunotherapy, with ranging from CAR-T to different anti-CD20 combinations or other monoclonal antibodies. The new bispecific T-cell engagers, as you know, that they bridge together the B cells and the T cells, and also immune checkpoint inhibitors. So we get into the preclinical models. So how are we going to test these new therapies? So as you all know, we all have experienced the simplicity of the cell line models and the complexity of the mouse models. And we have, in fact, very few percentage of patient-derived models in the preclinical models. This is something that has been developed in the last years. The cell lines have quite a few limitations. As you know, sometimes they represent more aggressive disease than they are. In the case of follicular lymphoma, which is indolent, it's very difficult to have follicular lymphoma cell lines that behave as the disease is. The microenvironment, if you want to do co-cultures, they are mostly allogenic. And of course, we don't have any patient heterogeneity in the systems. In the case of mouse models, either syngeneic, transgenic, or PDX, so of course we always have a problem of ethics and expenses. And with all this new regulation, at least in Europe, with the 3 Rs, this is something that we are reconsidering, and we are trying to reduce the animal testing to the last steps of the drugs validation. When we use PDX, which is a very useful tool, you know that they are immunodeficient. This is a very big limitation for immunotherapy, and you can have to use humanized PDX, which is complex and expensive. And of course, we are working with mouse, so this has limitations also in the context of physiology and in immunology, of course. So here we come with the patient-derived in vitro models that have some advantages. So these advantages, of course, are the recapitulation of patient heterogeneity. We have good cost-effective and short-term results comparing to animal models. We can include tumor microenvironment populations. We are trying to include autologous microenvironment. And of course, we can mimic this cell-to-cell interaction in these 3D structures. So I'm going to we'll share with you these patient-derived lymphoma spheres that we have developed for mantle cell and follicular. So what we do in this system is basically taking samples from our tumor biobank in IDIBAPS from the hematopathology collection. And we have samples from lymph node biopsies for peripheral blood. And these samples are basically PBMCs. So from the lymph node, we don't have really the whole cellularity in the lymph node. We have the result of mechanical dissociation. That means that we don't have macrophages or stromal cells. So if we want these populations, we have to reintroduce. So we are doing that by adding monocytes from Buffy coats from healthy donors. We have also done with autologous monocytes, if possible, when we have the sample, especially in follicular lymphoma. Then we mix these populations that are basically these purified monocytes together with the content of the sample, that is basically B and T cells. And we have optimized specific cocktails for each type of lymphoma, trying to recapitulate the signaling that we know is happening in each type of lymphoma in the lymph node. And we see it in these 96 well autologous attachment plates. So what I'm going to show you first is how we have characterized these systems, and then how we use them for immunotherapy testing. Okay. So starting with mantle cell lymphoma, this is the images that we have with these spheroids after about 7 days of culture with these cytokines that we have optimized. As you can see, the spheroids growth, gain volume. With the presence of cytokines, not with the presence of monocytes alone, and this also happens with the combination. But in fact, when we look at the proliferation, so it's just the cytokines what induces the proliferation of B cells, And this is also happening similarly with the presence of monocytes. In the cases of T cells, also the monocytes induce some kind of proliferation, probably because they were allogenic. So we have some kind of allogenic reaction. And this would be the summary of all the patients that we tested in terms of B cell viability. T cell viability, as you can see, is pretty good for a primary sample. We are about 70%, 60%. This is good after 7 days. Those working with primary cells maybe acknowledge this. And in terms of proliferation, so we are ranging from 30 to 40%. Really, these structures have no spheroids. They are more like pancakes, as you can see here by SPM microscopy. So they have this pancake structure, as you can see in these 3 images, about less than 1 millimeter. And they recapitulate the hallmark translocation, in this case, 1114. In terms of composition, so they basically B cells, T cells, and monocytes in this proportion. So about 10% T cells and monocytes and 80% B cells. CD4 and CD8 are more or less equally distributed, with more prevalence of CD4, which is also what happens in the lymph nodes. And then we wonder what happened with the monocytes we introduced. So the monocytes, we introduced monocytes because we did with macrophages, but what happens is that you don't have any extracellular matrix in the spheroid. The macrophages basically aggregate in the center and they die. So we thought that maybe we can introduce monocytes, and as we have done previously in 2D, we knew that these monocytes are able to differentiate to macrophages basically with the stimulus of the tumor. So because the tumor cells secrete some CSF1 and other cytokines that favor the differentiation into macrophages. So we assess what happened with these monocytes, if they differentiate, if they polarize to M1 pro-inflammatory or M2 anti-inflammatory and more tumorigenic. And what we found in this MCL-PDLS is that, as you can see here, the monocytes here in this size forward and side scatter plot, so they gain in size and in complexity in the PDLS, not to the level of a full macrophage, but certainly they engage some process of differentiation. When we look at M2 and M2 markers, as you can see here in red, so the PDLS express more M2 markers than M1 markers, meaning that these monocytes have at least partially differentiate to something that goes more towards M2 macrophages. When we did principal component analysis, as you can see here, so the MCL-PDLS macrophages, they are closer to M2 than to M1 macrophages. The next thing we wonder is what happened with these B cells that we have in the PDLS. They are coming from the peripheral blood. Do they resemble the transcriptome of those B cells in the lymph node of the lymphoma, of the mantle cell lymphoma, which is really what we want to mimic? So to address this question, what we did is we take this peripheral blood from the patients, we generated the PDLS, and then we isolate the B cells from the peripheral blood before doing the PDLS and after doing the PDLS, and we did RNA-seq. Then we wonder, we ask if this transcriptome really mimic the lymphoma, the lymph node transcriptome, using specific signatures that were already available, published by other groups. So what we first acknowledge is that when we compare the PDLS transcriptome with the peripheral blood sample, not without any stimuli, so we see that there is a great number of genes that have been upregulated and unregulated. So there are big changes in transcriptome. And then when we look at these genes, we really see that we get an increase and enrichment of those signatures that I mentioned before in terms of the lymph node signature or the proliferation signature, together with other canonical pathways that are known to be regulated in mantle cell lymphoma lymph nodes. So we were happy enough to see that these B cells really have a transcriptome closer to those in the lymph node of mantle cell lymphoma. We also did a bigger analysis, using genes and enrichment analysis, asking different canonical pathways, and we saw that there were a number of interesting pathways upregulated in this MCL-PDLS related with proliferation, immune response, addition, or angiogenesis. And these are representative enrichment plots. So as you can see, they're pretty good FDRs and normalized enrichment scores. The next thing we wonder is, are we really gaining something for culturing 3D? So as you can see by the numbers, we gain something, but not a lot. So basically the big work is done by the cytokines. So this is something that we were not so super happy to see that, but that was the reality. So in fact, there were few genes that were upregulated, but they were not, we cannot disregard this, because in fact, in 3D, we get an enrichment of those signatures that I mentioned before related with the mantle cell lymphoma lymph nodes. So something is happening extra in 3D. And also, it was interesting to see the upregulation of other interesting pathways, including MYC, stemness, and which also I like more is those related with the collagen and the extracellular matrix organization. So in fact, probably the presence of the monocyte macrophages so engage some process of extracellular matrix generation. So in fact, the 3D is not that bad, even though it's not the main component of this PDLS. The next thing we did is to look at the T cell compartment, because as I said, there's basically composed of B cells, autologous T cells, and allogenic monocytes. So to see what happened in this PDLS, we first compare the transcriptome of mantle cell lymphoma lymph nodes with normal tonsils from healthy donors using public databases to see which were the immune molecules that were really enriched in the mantle cell lymphoma lymph nodes. So what we get is that in mantle cell lymphoma lymph nodes, there are several pathways, immune pathways that were enriched, and whereas among them, we focus in TIM3 pathway that are in the interface between mantle cell and the T cells, including TIM3 or PD1, PDL1 or CD70 or TIGIT pathway. The same happened with CD47, for example, in the interface with macrophages. So when we look for the expression of these immune molecules in our PDLS, so we see that both in CD4 and CD8, we have a higher expression of these immune molecules compared with the initial samples, so getting overexpression of these immune checkpoints, and the same happened with the ligand in CD20, also in B cells and in monocytes. So with this, we concluded the characterization of the different actors of the PDLS, and we were pretty satisfied that they were somewhat close to the mantle cell lymphoma lymph node. Then we use this system to assess different types of immunotherapy and we wonder if they can recapitulate in vivo responses. So to do so, we basically generate the PDLS, and after 3 days, we did the treatments, and after another 72 hours, we analyzed the effect of the drugs. So we did these 2 points of analysis. So what we first did is we benefit from a collection of samples that were treated, that come from patients treated with ibrutinib at our institution, and where we knew the response in vivo. So we treat these samples, we generate the PDLS with these samples, and then we treat with ibrutinib, and we observed that those patients that were sensitive in vivo, so were responder patients in vivo, they also show some kind of response in vitro, while those patients not responding in vivo didn't have any response in vitro. In terms of viability, when we were looking at the whole B-cell depletion, because ibrutinib is basically doing more inhibition of proliferation than cell death. So when we really compute the whole number of B cells, we really see clear that those responding patients in vivo, they also respond in vitro, and the reverse happened with the non-responders. So we were able to recapitulate these clinical responses in vitro. And very interesting case was this one, was a patient treated in first line with R-CHOP. And in the first relapse, he was treated with ibrutinib, which is the typical scheme. And we were lucky enough to have samples pre and post ibrutinib when the patient relapsed. So we have samples sensitive and resistant to ibrutinib. So when we generate PDLS with these samples, we observe that we could validate that those samples, that those PDLS generated with a sample pre-treatment were sensitive to ibrutinib, while those PDLS generated with a sample post ibrutinib and thus resistant to ibrutinib was also resistant in vitro. Then, as I said, we have a big interest in testing immunotherapies. So we decided to combine ibrutinib with anti-PD-1, with nivolumab. And this is a combination that has done good has given good results in other lymphomas, such as DLBCL. So what we observe is when we combine ibrutinib with nivolumab, we didn't get any benefit, any significant benefit from the combination. But when we focus in those patients with limited responses to ibrutinib, in those cases, we really see an improvement, a benefit from the combination. When we focus in those patients that we know the response in vivo, so those patients responding to ibrutinib in vivo, we see the same. So those patients responding in vivo to ibrutinib didn't benefit from the combination, but those patients not responding did benefit from the combination, meaning that we have an option for those patients resistant to ibrutinib. What else? Something that is interesting, I think, to note, and is something we have seen in several models, is when we treat with immunotherapy, with immune checkpoints, we always get this interferon gamma secretion. But this doesn't correlate really with toxicity. In fact, what we see correlates with toxicity is the secretion of granzyme B and the increase in the percentage of CD8 T cells. So as you can see here, those patients sensitive to the combination really give this increase of the granzyme B and the percentage of CD8, but not in those patients that didn't respond to the combination. So with this, I have finished the mantle cell lymphoma results, and I will continue with the follicular lymphoma models. So in the case of follicular lymphoma, we did the same scheme of generation of the PDLS, and as you can see here, the results are similar. So with the cytokines that are different from between mantle cell and follicular, so really get a significant growth of the spheroid, and this is maintained with the presence of monocytes. In terms of viability and proliferation, so it's also pretty good. The proliferation is bigger in this system with the cocktail compared with mantle cell. And we get also this structure that are basically this pancake structure by spin. We did a confocal microscopy, and we see how they are mostly composed of B cells, interspeed macrophages, and T cells. And we were able to do, which is quite challenging, immunohistochemistry of the spheroids. And we were able to reproduce the expression of the typical markers that are done in pathology to characterize this lymphoma. That are overexpression of BCL2, basically expression of the B cell marker CD20, the germinal center CD10, and the proliferative marker Ki67. The T cells are mostly located in the interfollicular areas, and this happens here in the periphery. We also observe the maintenance of this hallmark translocation, as we saw for the mantle cell, so in this case, the 14-18. The composition is a little bit different, and it's nice because in the follicular lymphoma lymph nodes, the infiltration of T cells is higher, and this is what we get also with these optimized conditions, with more about 30% of T cells and less than 10%, about 5% of macrophages. This is something that we have observed. The monocytes are less happy with this in this system than in the mantle cell. This is something we still have to study. But these monocytes also gain complexity, as you can see here. So they differentiate to something that are close to macrophages, not with a definitive phenotype, more like an intermediate phenotype between M1 and M2, as you can see here, because they express both M2 and M2 macrophages. So we follow with the analysis of the B cell transcriptome, as we did for the follicular lymphoma. So we did analysis a little bit different. So in this case, we had the possibility of analyze the follicular lymphoma lymph node and the peripheral blood from the same patient. So what we did is we generate the PDLS with the peripheral blood, and then we isolate the B cells from the lymph node from the peripheral blood from the PDLS. And we wonder how these B cells from the PDLS resemble the B cells of the lymph node. So we did RNA-seq, and when we compare the lymph node versus the peripheral blood and the PDLS versus the peripheral blood, we observe a significant number of genes that were both upregulated in the PDLS and in the lymph node. And when we look for specific signatures, what we did is, in this case, in follicular, there were not already generated signatures for the lymph node. So we generated a signature of lymph node using, because we have pairs of lymph node and peripheral blood from the same patient. And in fact, we observe an enrichment of the lymph node signature in the PDLS we generated. Besides, when we did a broader analysis by GSA, so in the PDLS and in the lymph node, we have some interesting overlapping of different pathways enriched that are related with proliferation, MYC, apoptosis, metabolism, and immune response. And this is happening both in the PDLS and in the lymph node compared to the peripheral blood. We proceed similarly to mantle cell to see what happened with the T cell compartment. So we did a similar analysis, so taking public databases and comparing the follicular lymphoma lymph nodes with tonsils, and then look for those immune molecules that were upregulated in our system, that were basically the PD-1, PD-L1 axis, the ICOS ligand, the TIGIT axis, the TIM3, the LAG3, and the CD70. So then we validated these results by flow cytometry, as we did before. So we were able to see that all these molecules were upregulated in our PDLS. And something that we did here that we haven't done in mantle cell is to really study better the T cell compartment. The T cell compartment is very important in follicular. And in fact, we have more material because, as I said, there are more T cells in the system. We were able to demonstrate that the T cell compartment is composed of a spectrum of phenotypes, including naive, central memory, effector memory, and also effector TEMRA. And this is something that is acknowledged also in the single cell studies in follicular lymphoma lymph nodes. So it was something that was nice to see. We also see that the different subpopulations of CD4 cells are preserved: follicular helper, Tregs, antifollicular regulatory. And which is also more important, when we analyze the expression of PD-1, TIM3, LAG3, and ICOS, we were able to identify different subpopulations in CD4 and CD8 with different status of in terms of resting, activation, or exhaustion. So we can recapitulate like a spectrum of activation and exhaustion, like it has been described in single cell studies. So as I said, as we did before, so we follow with the usage of this system to test different immunotherapies. We started with testing several immune checkpoints. As we said before, we focus in those immune checkpoints that were upregulated in our system in follicular lymphoma. So we treated the PDLS with rituximab, which is the standard of care, and with different antibodies targeting TIM3, LAG3, TIGIT, PD-1, or ICOS. And as you can see here, the response as single agents of these immune checkpoints were very limited, with the exception of nivolumab, which is really the one that is in the clinic. And nivolumab, when we was combined with rituximab, so we have some increase in the depleting activity, but it was not significant. In fact, there were a portion of patients that were sensitive, but a good bunch of patients that were resistant. So what we did next is to try to investigate the causes of these different response. So we looked for the expression of different immune checkpoints, as we have done in the system, and we found that the TIM3 axis was related with these limited responses to both rituximab and rituximab nivolumab, meaning that those PDLS, those cases, because we were quantifying this expression of these immune checkpoints at different time points before the generation of the PDLS, after before addition of the treatments and at the end of the treatment. So we observed this correlation of TIM3 expression at the beginning of the generation of the PLELS in the CD8 and the expression of C66A, that is the ligand in the B cells at the end of the treatment, meaning that this axis has something to say to the responses to this combination. So we thought that maybe we can combine rituximab with TIM3 to get a good combination. And as you can see here, it was a little bit disappointing. So TIM3 was doing nothing, and the combination was even worse than the rituximab alone. And this was the way we separate by sensitive and resistant. In fact, there was one patient benefiting from the combination. But what we realized is that we have disregarded that TIM3 is not the only ligand. I mean, it has more, 66 is not the only ligand of TIM3. Galectin-9 is also a ligand of TIM3. So the problem is these antibodies against TIM3 is not blocking. They are not blocking the binding of galectin-9. So we try to combine with galectin-9. And we first, before doing that, we discovered something that was pretty interesting, that we quantified the galectin-9 in the supernatants of these PTLS, and we observed an inverse correlation of the galectin-9 in the supernatant with the response to the combination of rituximab with TIM3, meaning that galectin-9 was doing something wrong in the system. So when we combine rituximab with galectin-9, we really see a benefit compared to rituximab, meaning that really what we need to combine with rituximab is galectin-9 and not TIM3. And this is something we are now pursuing a little bit more because basically we stopped the studies here. And this is included in the paper we have now under revision. And the other thing that we did in this system that is pretty interesting is to use to test CAR-Ts. So in our institution, we have a CAR-T program, and they have developed several. Two are already approved for different hematologic malignancies that are CD19 called RE1 and BCMA called RE2. And they are now trying to get approved by the IMS. The RE3 product that means the combination of the BCMA and CD19. So at that moment, and with this collaboration, they wanted to know if it was better to do the co-transduction of the BCMA and the CD19 CAR-T, meaning that we have three products. So those CAR-T expressing CD19, BCMA, or those expressing both antigens, or generating a bicistronic, that means that both targets are in the same construct, so you have just the product expressing the two antigens. So what we did is we generate PDLS, and we treat with the different CAR-Ts, so the RE1, the RE2, the RE3, and the bicistronic. And as you can see, the four CARs induce changes in the morphology, but they are not the same toxic. And as we look for the specific lysis, the more active were the CD19 alone or the RE3, so meaning the co-transduction product, but not the bicistronic. And we really, when we do extended incubation, we really see a benefit from the co-transduction product, the RE3, comparing to the single RE1 CD19. And this is also seen when we look for the secretion of different molecules that are typical in the CAR-T studies, that are interleukin-2 for the proliferation of T cells and TNF-alpha interferon gamma. And it was also more interesting when we were looking for with a CD19 negative case. In this situation, it is very clear the benefit of the RE3 because the RE1 is not active at all because we don't have the antigen, and the RE3 is clearly the option in these cases. Okay, so just to finish, I will dedicate a couple of minutes to introduce the last model we are working. So beside the spheroids, we need to introduce adherent cells, so meaning that we want to introduce stromal cells, macrophages. So we need to introduce some type of extracellular matrix. So to do so, we are using collagen 1 from rat. And in this system, we are introducing follicular dendritic cells that are derived from tonsils, and we have modified to express germinal center cues. So in this case, what we are doing is to, instead of adding the cytokines soluble, we have the dendritic cells that express most of the ligands that the follicular lymphoma needs. And this is something basically mimicking that what we see in the follicular lymphoma lymph nodes, because in the follicular lymphoma lymph nodes, we have these structures, these dendritic networks that are packing the lymphoma follicles. And this is what we get here. So as you can see, we have these nice structures packaged by the dendritic cells, and inside you have the tumor cells that are proliferative and happy. And we have in this system, the proliferation of T cells is more compromised. This is something that is different from the other. In terms of B cell viability, we have very good viabilities and also very good proliferation that is increased when we have this, that is obtained, we will have these dendritic cells in the system, and is increased when we also introduce macrophages. And something that is happening that is also quite interesting is in the germinal centers, we have this isotype switching of the immunoglobulins, and this is something that is happening also in our system, meaning that the germinal center is happening. So this is something good. We have initially IgM, and we are getting IgG after the generation of the tumoroids. We are characterizing this system. First, we have done some morphological, structural characterization by electron microscopy, trying to characterize the size of the pores and to see if they are close to those in the lymph node. We have characterized the viscoelastic modulus, the stiffness, and as you can see here, so the collagen has a very alone has very low stiffness, but when we introduce the tumor cells in green, or the tumor cell with dendritic cells plus the macrophages, they are in the same range as the lymph node. And also something interesting is even we are using this collagen ground from rat as a scaffold. The system is also capable of secreting their own extracellular matrix. So we have been able to see an enrichment in collagen 4, tenascin, and fibronectin, and others that we haven't validated yet by immunofluorescence. And we know that these extracellular matrix proteins are overrepresented in follicular lymphoma lymph nodes. So this is something good that we are also able to see them in the tumoroids. And to finish, I also can tell you that we have used this system to test CAR-T, the same as I mentioned before, the RE1 and the RE3. The CAR-T are able to get into the collagen, so there is no problem about that. We get different responses, and we also see the typical case that is not responsive, but this case is negative, as we saw before. And in this case, is really very convenient to use the RE3 that I just mentioned before, that has this double antigen recognition of CD19 and BCMA. And with this, I would like to finish and just some concluding remarks to convince you that we have several advantages of these patient-derived models, including that they are, of course, recapitulating the 3D biology, the patient heterogeneity, the inclusion of microenvironment populations, and of course, it's a cost-time convenient option. Regarding the spheroids, I think it has been clear that we can recapitulate proliferation and viability, both in tumor cells and in autologous T cell. This is one of the good things of the system, to have the autologous T cells. We can recapitulate transcriptome in terms of transcriptome of the B cells relevant pathways that are important in the lymph node. We can recapitulate the T cell immune exhaustion profile that we have in the lymphoma lymph nodes. And we are able to reproduce these clinical responses I mentioned, and to discover novel immunotherapeutic combinations that may have an opportunity, especially for those resistant patients. And the beauty of this tumor that I just explained is the possibility of integrating stromal cells. So the system is more rich. It's also less easy to manipulate, in the sense that when you have to use the system for flow cytometry, so it's a little bit more painful. But of course, I think it's a superior system. Having this extracellular matrix, and it's also the base that we are using to integrate in microchip or microfluidics to introduce vascularization and so on. So I think this is the way to go. And with this, of course, I haven't done all this work. There's a bunch of nice people in my lab doing this work: Ferran, Celia, Juan, Neus, and Ruben, and many collaborators from Toulouse, because this project was done in the context of an Lympho Interreg grant. And also the people in the IMACTIV, we did the 3D studies, and many other people in the hospital. And thanks to you for your attention. Happy to answer any question.
Thank you, Patricia. I think it was really interesting. I think Tony has a question. So if you want to directly ask Patricia.
Great. Yeah, that's great, Patricia. I have a question. What is the two questions there? What is the source of the T cells in your, I think it was the checkpoint blockade experiments with nivolumab? Is it just the ones that come along with the follicle or the lymph node, or are you adding extrinsic T cells?
Yes, they are just the ones coming in the sample. We are not adding T cells, external T cells. The only external stuff is the monocytes, that we are not really quite happy about that, because even though when we have done really studies comparing allogenic and autologous monocytes, and in terms of B cells, the transcriptome is not really suffering, but we haven't really studied the T cells, but I'm sure the T cells are getting crazy. So it's something we are working on that. Yeah.
And if I can follow on to that, another immunology question is that you are at least former participant in the cell death field, and hopefully again will be. So let me ask you, on your immunology plots, when you're looking at the elimination of cells, you describe it as specific lysis. Can you describe how specific lysis is distinct from cell death, and why immunologists have to call it something completely different?
Specific lysis, okay. So you basically use aqua or some like a live proof. So there is no identification of apoptosis or anything. So basically you have the cell as a whole, or it has been destroyed, because the problem with the CAR T is they basically explode the cells, so they are not anymore there. It's pretty.
Patricia, we'll have to talk longer about this. They don't explode the cells. It's programmed cell. It's programmed cell death. Like there's no magic involved. It's programmed cell death.
It's programmed cell death. Okay. So, but we don't do phosphatidyl setting or anything. We basically use aqua cell trackers. So for viability.
Totally legit. I just never understand why it's always labeled specific. Like it's a different magical event.
No. I'm gonna tell you. No. I'm gonna tell you. Let me explain you. Specific lysis because you subtract the lysis induced by the T cells untransduced.
Lysis.
Yes,
but
listen.
It's not lysis.
Yeah.
It's just
it's not lysis. Okay, it's not lysis. You're right. But anyway, it's taking out the background of the allogenic T cells, because your CAR T are allogenic.
Yes,
so it's basically this. But the lysis, okay.
Specific killing.
Okay, specific killing. Okay.
I have to agree with Tony on this one. I'm sorry, but I'm gonna continue the discussion.
Okay.
We have a question from Russell Jenkins.
Yes, so wonderful talk, very exciting. I had one question that's a little bit more technical from the first part of the talk. And it was relating to the cytokine cocktail that you used. And I'm sorry if I missed the particulars, but are you able to share what it is?
Sure. It's published. Yeah, sure. It's published. For the mantle cell, it's basically CD40 ligand, interleukin-4, and BAF, I think. Yeah.
All right.
And for the follicular, we are about to publish, I hope, but maybe you have to wait. So okay. It's under revision in Blood Cancer Journal.
No,
but it's basically it's mimicking the T cell interaction and some cytokines that we know that are in the lymph node. That's it. There is no more secret. Yeah.
All right. So it was rationally designed for the specific.
Sure.
And then
we try several. Huh?
All right.
Quite a few.
That's the backstory. Then from some of the molecular pharmacology work with the ibrutinib and whatnot, are you able to share a little bit more how you arrived at the concentrations of the drugs you used, and how much kind of drug testing and dose responses and time courses you're able to do in this model system versus needing to rely on some of the more established cell line models to do some of that work, and then pick the dose and then go back to the PDLS, etc.
I mean, in fact, as I said, it's a 96 well-played system. So it's we are basically using triplicates or 6 replicates depending on the material we have, but we are able to do several doses and several time points. This is not a problem. For the ibrutinib, we were using doses that we knew for the 2D studies. So, and from the monoclonal antibodies for the immune checkpoints, we were also looking for some publications and using 2 or 3. So we didn't do really a dose response, but we were basically going up and down of the published data.
Thank
But
you.
it's not a problem really to do, to include several doses in the plate. Thank you.
So we have a question from Terry. With such large structures, hundreds of micrometers, are you concerned about nutrient and gas exchange reaching the cells in the center of 3D structures? And do you expect those conditions to change the transcriptome, for example, the biology of the system?
Yes, so this is something we wondered also, because we also thought that maybe in the center, maybe some hypoxia. And in fact, there is some cell death in the center. Yeah, that happens. But in general, the structures are not very compact, at least for the spheroids. For the tumoroids, that may be that may be something to explore. In fact, we are now we have done single cell for the tumoroids, so we are analyzing. So we'll see. Maybe we get some hypoxia. Yeah, good point.
I have one question too. So you mentioned especially for the PDLTs that you use collagen. Have you tried other synthetic hydrogels? Like I don't know, they're like the ones that come to my mind are BioGel X, that they have like different structures. So basically you can combine them. Yeah.
A disaster. The BioXL, a disaster. We try BioXL. We try some other types of collagen. We were also working well. We tried jellyfish collagen, and it was pretty good too. But we have some problems with the source, because they have problems with the production. And in the end, the easier thing was the rat one. And in fact, rat one you can prepare yourself. We haven't done, but there are people generating, their own batches, and it's convenient, and cheap. Yeah, we suffer a lot with the pandemics, with the the matrigel people were suffering, the collagen people were also suffering. I like collagen compared to matrigel because it's cleaner. The matrigel is full of stuff that we don't want. And in the end, this collagen is just the scaffold that allows you to generate other, as I said, extracellular matrix. That is the one in the end that you're interested in.
Very interesting. We have another question from Sonam Bhatia. She says, "It's super exciting work. Thank you. I'm wondering if you can comment on some technical factors with culturing these PDLS. What is the success rate of making them from patient samples? What is the maximum amount of the time that you can keep them in culture? Or are these primarily short-term cultures?"
So, I mean, depend basically in the variability of the initial variability of the sample. I mean, with the cocktail that we use, we make miracles, but still, if the initial conditions are very bad, you cannot recover. For the time, it's basically up to 10 days. More than 10 days, we haven't tested. That's the reality. Yeah, we haven't gone farther than that.
And I have another question too. So do you think that you can expand so that you can use these models, the PDLS or the PDLTs, types of cancer, for example, other hematological malignancies or even solid tumors? Do you think that it can be?
Sure. I mean, for the PDLS, we have done I haven't explained, but we are setting up for the LBCL and also for marginal zone lymphoma. So this is something that in the end, you have to come with the right cocktail that you are satisfied and that you are able to reproduce what you want in terms of B and T cell transcriptome. But I think that, it's a very simple system in the end, but it's convenient for replicates and for handling, because you can get flow cytometry very easily. With the imaging, you cannot get really the reality of what is happening inside. So they are very convenient for that. When you get the matrix in the system, is very nice for to get a more relevant system, but it's a pain really to analyze. And also when you have to get to analyze the B cell depletion, you always have this bias for the process of disgregation. Even so, you always do the same, very consistent. So you will have doubt that, are we recovering the same? Are we incubating the same with the collagenase, the space, and so on? So the replicates is much more consistent with the spheroids, with all the limitations that the system have, of course. But I think this is superior as the previous to the system local cultures. I think something that has something to add. And in fact, I mean, with this system, we have a collaboration with Roche for immune checkpoints for follicular and marginal zone. Yeah, so this is something that I think people from the companies are starting to trust.
Interesting. We have another question from Elliot. Very interesting. Thanks very much. I missed this maybe, but what is the technical issue with getting monocyte macrophages from the biopsies?
Oh, yes. So the problem is, I mean, from the peripheral blood, you basically don't have enough. So this is not representative of what you have in a lymph node. But from the lymph nodes, we are in the clinic, so we are not working on that. But basically what they do in the hematopathology unit where the lymph node arrives is to disgregate mechanically, no enzymes. So you just get the PBMCs, so anything that is not attached. So you don't have any macrophage release or any dendritic cell or anything. So that's the problem. Now the problem is also the amount of sample that arrives to the hematopathology section. We are not working on that, and we have a new circuit thanks to Elias Campo, to the super boss. We are trying to get a piece of lymph node for us. It will make a difference. Yeah.
Well, I know that this can be complicated.
The primary material is always complicated. Yeah.
Exactly. I think that we don't have more questions. I would like to thank you, Patricia, for this excellent talk.
Thanks to you.
And thank all the members that joined us today. So I'll see you all next month. Exactly.
Likewise, I just wanted to say, Patricia, that was really, although I barged immediately into a criticism of like some tiny little item of nomenclature, really I shouldn't have overlooked what a really beautiful system you have for the study of lymphoma cells. Nice job.
Thank you very much to all of you, and I hope you have learned something about our model. Happy to collaborate and to talk any other time. Thanks for the invitation. A pleasure. A great pleasure.
Okay. Thank you all. Thank you.
Bye-bye.
Bye.
Bye, Tony. Bye, John. Bye.